In Chapter 7, we are going to learn how the nutrients that come in from the digestive system actually get used to release energy. So we know that energy comes from food. So energy obtained by breaking down different molecules. So the foods that we eat, and ultimately, all of that energy originates in plants through photosynthesis. So all of our energy comes from the sun. Plants convert it into usable sources, and then we can digest that food and get energy from it. So this process of cellular respiration is how energy is released from those organic molecules. And mainly what's happening is the stripping away of electrons from those molecules in order to make ATP. So electrons get released from the molecules like glucose or fats, and then those electrons can be used to make ATP, which is our energy store. So this process of cellular respiration, so respiration, we're just kind of talking about gas exchange. So we have oxygen coming in, carbon dioxide going out. This is why we need to breathe in oxygen through our lungs, and this is why we exhale carbon dioxide as a waste product. So again, the majority of eukaryotes use or harvest all of their energy using these electrons. So our reactants, which are everything on the left-hand side of this arrow, so this is glucose. Again, we just mentioned oxygen. And then the products, which is everything to the right of our reaction arrow. So it produces carbon dioxide, which is exhaled, releases some water. But again, the main point here is to release energy in the form of ATP and also heat. Now, again, we were talking about using electrons, so we need to talk a little bit about this process of oxidation and reduction, which is how electrons get passed from one molecule to another that ultimately can generate this ATP. So the passing of electrons through oxidation, this is when an atom or molecule loses an electron. So oxidation is the loss of electron. So we oxidize glucose to release its electrons. Those electrons have to go somewhere, though. They can't just hang out in space. So whenever there's an oxidation or loss of electrons, there's also a reduction, which is the gaining of an electron. So glucose gives up an electron to another molecule. So glucose is oxidized. The molecule it donates to is reduced. Because these have to happen together, again, the electron can't just be in space. We call these redox or oxidation reduction reactions because, again, if one molecule is oxidized, another has to be reduced. So these redox reactions, typically oxygen is the final electron acceptor. So again, we can kind of thinking about like hot potato or relay race or something like that. Those electrons get passed from one molecule to another, to another, to another, until finally there is something taking the electrons away. So we call that an electron acceptor. And in aerobic respiration, that electron acceptor ultimately is oxygen. That's why oxygen is needed for respiration. It's the final electron acceptor. Now the energy, again, follows the electrons. So the reduced form of an organic molecule has a higher level of energy than the oxidized. So looking at our little figure down here on the left, so again, oxidation, so our molecule A here is giving an electron to molecule B. So A is becoming oxidized. It is losing the electron, so it has a positive charge. Remember, electrons are negatively charged. Then molecule B becomes reduced because it has gained that electron. So essentially what is happening here, let me erase all of these molecules, all of these lines here that are going all over the place. But again, ultimately what is happening in this oxidation reduction is the electron from A is being given to B. So that causes A, which is kind of shown here. So then A is becoming oxidized, B is becoming reduced. Again, the real important thing though here, we're talking about energy, is that energy follows the electron. So A that is oxidized is now at a low energy state. It doesn't have much energy. B that accepted that electron is now at a high energy level. All right, so as we look now at kind of an overview, and then we'll go through each step. So this series of, or this process of cellular respiration happens in multiple stages. So the first stage, we're going to start first by talking about glucose, but we'll also talk about how other molecules like fats or proteins can be used to get energy as well. But we'll start with glucose. So glucose, again, gets oxidized, the electrons are coming off. It is going to be converted into a molecule called pyruvate. And the process of doing that is called glycolysis. So glycolysis is the first part of cellular respiration. It releases a little bit of ATP, but it also releases some electrons. And we'll talk more about this NADH, but that's what's picking up the electrons from glucose. So those electrons are coming off of glucose and going to make NADH. Now, the pyruvate that is left after glycolysis, it goes through a process called pyruvate oxidation. Again, that means electrons are being released, and those electrons are used to make more NADH. We're also going to get the release of a carbon dioxide molecule, and the final molecule here is acetyl-CoA. Now, the next part of cellular respiration, that acetyl-CoA then enters what's called the Krebs cycle. More oxidation is going to happen, so more of those electrons come off to make NADH. More carbon dioxide gets released as that molecule of acetyl-CoA is further broken down, and we get a little bit of ATP here. There's another electron acceptor called FADH2. It also picks up some electrons. So everything over here, these are all electron carriers. They're picking up all these electrons being released through those oxidation reactions. And then ultimately, the majority of ATP is made when all of these electrons come down to the electron transport chain, and it harvests the energy from the electrons to make ATP. Now in the process, those electrons have to go somewhere. So oxygen is there to pick up the electrons, and that's why water is made. And now we have these oxidized forms of the NAD and FAD. They can now go back to pick up more electrons to form NADH and FADH2. All right, so just breaking this down a little bit. Again, we start with glycolysis. So this is happening in the cytoplasm. So a special organelle is not needed. So bacteria can go through this. Any cell can go through glycolysis. So it happens in the cytoplasm. Again, no oxygen is needed in this step to make the ATP made in glycolysis. Now, as that pyruvate that's made, if it gets further oxidized through cellular respiration and ultimately goes to the Krebs cycle, which is in the mitochondria, then this is where a lot more oxidation is going to happen. So we get oxidation to release all these electrons, and then ultimately those electrons go to those electron transport chains to make ATP. So the more electrons we have, the more ATP that is getting made. All right, so then coupling these reactions. So in glycolysis, it's a biochemical pathway, just meaning that there are multiple reactions. It's not just one step from glucose to pyruvate. It's a series of steps. All of them are catalyzed by an enzyme, so each reaction has a different enzyme. But ultimately, the glucose, which has 6 carbons in it, it gets split in half or broken down into 2 3-carbon molecules called pyruvate. So the breaking of those bonds is what produces the energy, and that energy is used to convert ADP to ATP. And the process of making ATP this way is called substrate level phosphorylation, where the phosphate is just given from one molecule to ADP to make ATP. We'll talk about oxidative phosphorylation in just a minute. So in addition to that ATP that's made, again, electrons, we have oxidation, so electrons are being released. They are picked up by NAD to form a molecule called NADH. So if we go back to our terms, NAD plus is the oxidized version. It doesn't have any electrons, so it's low energy. But as glucose is oxidized and electrons come off of glucose, our NAD picks them up, and it forms the reduced form, the high energy form called NADH. Again, NAD is just a carrier molecule. It picks up electrons and it gives them to somebody else. It serves really no other purpose than that. And again, this is pretty much the main way that organisms can keep making energy even if they don't have oxygen. For example, our muscles use this. If we are exercising and our lungs can't give us enough oxygen for all of our muscles, some of our muscle cells can switch and just go through glycolysis. So we don't need any oxygen for that. So we're still getting a little ATP as we break down and use up that glucose, even though no oxygen is around. And then some organisms like bacteria, they only go through glycolysis. That's all that they do. All right, so this is just kind of the overview here of glycolysis, so you can kind of visualize it. Again, we start with this 6-carbon, shown as these gray circles of glucose. Again, it gets split or we add 2 molecules of ATP. So we kind of phosphorylate or add phosphates. So we put 1 on each end. Those came from this ATP up here. So we phosphorylate it. So now we have this phosphorylated 6-carbon molecule. Then what's going to happen is we're essentially just going to break that molecule right in 2. And then that creates 2 3-carbon molecules. But they both still have this little phosphate that we just added onto it. So now through a series of reactions, we're going to release those phosphates and release some electrons. So we're going to go through oxidation. So both of those 3-carbon molecules ultimately are going to form the 3-carbon pyruvate molecules. While they're doing that, we get oxidation to make some NADH, and we get some production of ATP through that substrate level phosphorylation. All right, so we say that glycolysis kind of happens in 3 parts. The energy requiring or priming reactions. And again, it's called that because it requires a little energy to get started. So it needs 2 ATP to get started. Then we have the cleavage reaction where we split the molecule into 2 3-carbon molecules. And then we actually release the energy. So the energy harvesting. That's where the oxidation and the substrate level phosphorylation ultimately produce the NADH and ATP that is formed. All right, so then if oxygen is available, then there are a couple steps to finish oxidizing that pyruvate. So first, pyruvate is converted to that acetyl-CoA molecule, and then the acetyl-CoA molecule goes through the Krebs cycle, and by the end of the Krebs cycle, it's been completely oxidized. So all the electrons that started in the glucose have been released, so they can be used to make ATP. So the first step is called, or the oxidation is pyruvate oxidation. It is sped up or catalyzed by an enzyme complex called pyruvate dehydrogenase. But essentially what happens is one of those 3 carbons, one of the carbons comes off. So as the carbon leaves, it produces a carbon dioxide molecule. In addition, again, there's oxidation. So hydrogens and electrons are released from that. Again, the electrons have to go somewhere. So the electron from pyruvate is given to NAD. And then once it's reduced, once it picks up the electrons, it's now NADH. Now those remaining 2 carbon fragments, because again, one came off to make carbon dioxide, the 2 carbon fragment that's left joins with a cofactor, a helper enzyme called coenzyme A, and that ultimately produces acetyl CoA. So the 2 carbon fragment is acetyl, and then that coenzyme A that was added gives us the CoA. Right, so just again kind of visualizing this, so that 3 carbon pyruvate, one of the carbons comes off, and that produces carbon dioxide. Then we get oxidation, so electrons are going to come off. That's going to be donated to NAD to make NADH. Now we have this 2 carbon fragment that's left over, so coenzyme A joins with it, so that we ultimately end up with acetyl CoA. Now I said we would talk more about how other molecules besides just glucose can be used to make energy, and this is showing one way that they all kind of combine. So proteins, like our amino acids, could also be broken down to make acetyl CoA. Our lipids, like our triglycerides or fatty acids, those can be broken down to give us acetyl CoA, or again, those fats or triglycerides can also, or sorry, what this bottom part here is showing is that acetyl CoA, regardless of where it comes from, whether it's from glucose, proteins, or lipids, that acetyl CoA has 2 options. If we have enough energy, then ultimately it gets stored as fat. If we need energy, then it's further broken down in the Krebs cycle to give us ATP. But this is the reason why, even if you have a low-fat diet, if you eat more than you need, you take in more calories than you need, whether it's through protein or carbs, if you have excess amounts of acetyl CoA, that is going to get stored as fat. Only if you need energy is that acetyl CoA used for ATP. So again, our fat stores can increase if we're consuming more than we need. All right, so this is just kind of looking a little closer at NAD. So this transferring of electrons, this oxidation reduction. So again, here we have an enzyme in purple because this is all enzyme mediated. So a substrate, the starting material, comes in. It has some hydrogens and electrons. So like glucose, for example, has hydrogens and electrons. It comes in and attaches to one of the active sites on this enzyme. But then there's another site where NAD, which is a coenzyme, it's a molecule that helps an enzyme do its job. So NAD also attaches along with this substrate, and then we get that oxidation reduction. Electrons leave the substrate and they're picked up by NAD. Now our substrate has been oxidized and NAD has been reduced. And again, the energy goes with the electrons. So NADH has a higher amount of energy than what's left of the substrate. Right now, enzymes can be used over again, and now that we've changed them, the product, which again is the oxidized form, it gets released, and now NADH is free to take its electrons and hydrogens to be used in the electron transport chain. All right, so our acetyl CoA, as I mentioned in the a couple slides ago, that acetyl CoA that's made depends on the availability of ATP. If there's not enough ATP, so we need energy, then the acetyl CoA goes into the Krebs cycle to make ATP. However, if we already have enough ATP, then that acetyl CoA is used to make fat or triglycerides. And that's why we don't want to take in more than we need. All right, so in our Krebs cycle, again, this is occurring in the mitochondria, multiple reactions that are taking place. But again, ultimately, it is more oxidation of that acetyl CoA. So the acetyl CoA comes into the cycle. Remember, it has 2 carbons. It's going to combine with a 4-carbon molecule that was already there, and that gives us a 6-carbon molecule. Then through a series of oxidation reactions, 2 of those carbons get removed. That forms more CO2. But again, the point here isn't to make CO2, it's to get those electrons. and those electrons are picked up mainly by NAD, but there's also another electron carrier called FAD, and those can be used to make or to carry electrons. All right, again, we get a little bit of ATP made. Now we're left, once those 2 carbons come off, now we're left with another 4-carbon molecule. Essentially, it gets recycled back to the original 4-carbon molecule that was there at the beginning. But during that conversion or recycling process, more electrons come off, so more oxidation, and then NAD picks up some, and FAD picks up others. And FAD works pretty well the same way. It's just an electron carrier. It picks up electrons and gives them to something else. Right, so our visual here, the Krebs cycle, again, our 2-carbon acetyl CoA comes in. There's a 4-carbon molecule already there. And so those 2 join together to give us a 6-carbon molecule. Now the coenzyme A that was there, it just goes back and can keep being reused again. Right, so now this 6-carbon molecule is going to be oxidized, and one of the carbons comes off to make CO2. Again, oxidation occurred, so we get some NADH being made. Now we're down to a 5-carbon molecule, and then we release another carbon to make carbon dioxide. More electrons are released to make NADH, but we do also get a little bit of ATP made during that step as well. Now we're back to a 4-carbon molecule, but it's not exactly the same as the one we started with. So a few more reactions are going to convert the 4-carbon molecule made here back to the 4-carbon molecule that we started with. And this is also why this is called a cycle, because one of the molecules, this 4-carbon molecule that was at the beginning, gets remade, so it's a cycle. It just can keep going on and on as long as acetyl CoA is present. Again, during this recycling, though, we get some more oxidation, so FADH2 and NADH get made. All right, so by the time the Krebs cycle is completely over, from 1 glucose molecule, the cell has gained 4 ATP. It received 2 from glycolysis and 2 from the Krebs cycle. It has 10 NADH molecules. It got 2 from glycolysis. It got 2 from pyruvate oxidation, and it got 6 more from the Krebs cycle. It also received 2 FADH molecules from the Krebs cycle as well. So again, the main point by the end of the Krebs cycle, we haven't made a lot of ATP, but we have all of these electrons. So now we're ready to make the majority of that ATP. So NADH and FADH2 transfer their electrons to a series of reactions in the mitochondrial membrane. And this is why it's folded, because it allows more electron transport chains to be added. And that series of molecules is referred to as that electron transport chain. And this is just kind of a series of proton pumps, which are hydrogen ions. So they're moving those, and they're going to pump protons using energy from the electrons. Again, pump implies we're going from a low to high concentration, so it needs energy. And that energy comes from the electrons, and ultimately pumping those hydrogen ions or protons out into the inner membrane space of the mitochondria. Again, ultimately the electron has to be picked up and it has to go somewhere. So that is the job of oxygen. This is why we have to have oxygen, is to pick up those electrons. The electrons are negative, so it attracts some of those positive hydrogen ions, and ultimately those join together to form the water. Right. This process of using energy from electrons to make ATP is referred to as chemiosmosis. So as the concentrations of the protons build up in the inner membrane space, again, we're pumping them from the matrix using energy from electrons out into this intermembrane space. They want to go back into the matrix from a high to low concentration. They can't cross the membrane on their own, so they're going to enter through ATP synthase channels, which are transport proteins but it also ends with ASE, which kind of indicates that it is an enzyme as well. And as the name implies, it helps to make ATP. So the passage of those protons ultimately is what forms ATP. Right, and again, kind of our visual here. So these are the different complexes. So there's 1 here, 2, 3, and 4 of these proton pump complexes. So they're embedded. So the combination of all of these make up one electron transport chain. And ultimately, what is going to happen, let me get rid of these marks here. Ultimately, what's going to happen is NADH that came from glycolysis. So remember our little figure over here. So glucose gave us some NADH. The pyruvate oxidation to acetyl-CoA gave us some. The Krebs cycle gave us a lot. So all of these NADH and the FADH2 come to these electron transport chains. So the NADH gives its electrons to the first part. As the electron moves through this complex, it releases energy, and that energy is used to move these hydrogen ions across from the matrix, which is all the way inside the mitochondria, into this intermembrane space. Right, now that electron has to keep going, so it moves along to the next complex. Again, through this series of reactions, it releases energy, and that energy is used to move those hydrogen ions out into the intermembrane space. Again, our electron has to keep moving. It can't stay there, so it goes to the final complex. Again, more energy is released, so more hydrogen ions come out into that intermembrane compartment. Finally, our electrons have to go somewhere at the end of this complex, so that electron gets picked up by oxygen that attracts some hydrogen ions, and it makes water. So water gets made when oxygen picks up those electrons at the end of electron transport. Now, that's not the point. What we're focusing on here is setting up all of these hydrogen ions. So they're concentrated now in the intermembrane space. They really want to come back into the matrix, right? And whenever they do that, it forms that ATP. All right, so then what happens whenever cells don't have oxygen? If they can't remove their electrons from electron transport, they can't go through pyruvate oxidation or the Krebs cycle or electron transport. So in the absence of oxygen, cells can go through a process called fermentation. So instead of further oxidizing after glycolysis, what happens is that NADH molecule made by glycolysis, it donates its electrons to an organic molecule. So instead of oxygen being the terminal electron acceptor, some organic molecule picks those electrons up. So NADH gives its electrons, we'll see a better picture, to an organic molecule. That allows NAD to go back to glycolysis, pick up more electrons, and keep glycolysis going. Because our cells only have a limited amount of NAD, so it has to keep donating its electrons. If it can't donate them to electron transport chains because oxygen isn't present, then it gives them to some organic molecule in this process of fermentation. Right now, bacteria, many of them, this is their way of getting energy. They don't switch between aerobic and fermentation. They just do fermentation. They go through glycolysis, they get some ATP, and then they go through fermentation to recycle their NAD. So there are all kinds of different types of fermentation. Now, eukaryotic cells like our own, some of them are able to do a couple different types of fermentation, so we can switch between aerobic respiration when we have oxygen and fermentation when we don't. And this is showing just a couple of all the ones that are out there. The first one is ethanol or alcohol fermentation. So yeast, which is a eukaryotic fungus, for example, go through this type of fermentation. Now, of course, we use it commercially to make alcohol for us. But the yeast itself, they're just getting energy. Their end goal is not to make ethanol. They're just trying to get ATP. So what happens here in ethanol fermentation, again, glucose goes through glycolysis, makes those two pyruvates, but we also have some NADH, and then it has some ATP. This is the energy that is received from glycolysis. So it doesn't get a whole lot compared to aerobic respiration, but we do get a couple for each glucose. Right? Again, though, eventually glycolysis would stop if the cell runs out of NAD. So the NADH made in glycolysis donates its electrons to a molecule called acetaldehyde. Now, it was made whenever pyruvate from glycolysis gives up a carbon to make carbon dioxide. That gives us a 2-carbon molecule called acetaldehyde. Okay, well, it is actually what gets the electrons from NADH. When those electrons are added to acetaldehyde, it produces ethanol, which is alcohol. Again, not the goal for the yeast, but what is made that the yeast need, they need those NAD molecules to go back to glycolysis, pick up more electrons to make more NADH so they can keep making ATP. But again, we harvest that ethanol from yeast in order to make alcoholic beverages. All right, now our muscle cells, again, we can switch between aerobic respiration and fermentation when we don't have oxygen. And the type of fermentation we do is called lactic acid fermentation. So we don't make ethanol during fermentation. Instead, we make a molecule called lactate. And it's very similar, but actually one little step easier. So again, glucose goes through glycolysis, makes the 2-pyruvate. We get those 2 ATP molecules. That's all the energy we're going to get. But again, that NADH, those electron carriers, have to give their electrons to something. So in the case of lactic acid fermentation, those electrons go directly to pyruvate that was made. Now, whenever you add in all those electrons, it's no longer pyruvate. So the molecule made there is called lactate. So it still has its 3 carbons. No carbon dioxide gets released during this process. But again, the point here isn't to make lactate. That actually is an acid, so it builds up. That's why our muscles get sore. If you haven't exercised in a while, is a buildup of that lactate. But again, the point here is to give these NAD molecules that can go back to glycolysis, get more electrons, and we can keep going through glycolysis to get more and more energy. All right, again, as I said earlier, we started with glucose, but that's not the only food that we use. We can also get them from other sugars. So thinking about like our disaccharides, like lactose or table sugar, or even our complex carbs, like cellulose. Well, we can't get energy from cellulose because we can't break it down, but starch or glycogen, we break them down to get the glucose. But in addition to the carbs, proteins can also be broken down into amino acids. And then amino acids can be converted to different molecules that go to the Krebs cycle. Then our fats or triglycerides, the fatty acids can be released from those fatty acids. And through a process called beta oxidation, again, releasing electrons, those fatty acids are converted ultimately to acetyl-CoA. And that goes into the Krebs cycle, releases a lot of electrons through more oxidation. Those electrons all go to electron transport, and we get lots of energy. All right, so just a nice little summary here. So again, our polysaccharides like starch or glycogen, or different sugars like sucrose, ultimately glucose gets released. It goes through glycolysis to give us pyruvate. Pyruvate gives us acetyl-CoA, goes through the Krebs cycle to give us electrons that ultimately yields all that ATP. All right, but as this is showing, different lipids or fats can be broken down to release the fatty acids. Then through a process called beta oxidation, those carbon, like those chains of carbons, can be broken down to make acetyl-CoA, and then it goes to the Krebs cycle. The Krebs cycle doesn't know where acetyl-CoA came from. It's just oxidizing it to release all of those electrons. And then finally, our proteins can be broken down. We release the amino group, so that deamination that releases ammonia. This is why people with a high protein diet, their urine may smell kind of strong because of all that extra ammonia. Or even in individuals with diabetes where they're not breaking down carbs because they can't take in glucose, their body switches to breaking down proteins, so they also have that strong smelling kind of ammonia smell. Anyways, what's left, so those amino acids then, they can kind of come in a few different ways because there are 20 different amino acids. They all kind of hit different places. So some can become pyruvate, some do become acetyl-CoA, or some just come into one of the other parts of the Krebs cycle. Again, ultimately it gets oxidized, makes some NADH, and that goes to the electron transport. We're not really going to talk much about nucleotides because nucleic acids, we don't typically use those for energy. We use those to make our DNA or RNA for genetic information. But they could be broken down into parts, to inner parts of the Krebs cycle to give us some energy. Right, and that wraps up chapter 7 for us.